Today I would like to share with you some of our recent work on repurposing technologies that are already readily available and about to be outphased, you can say. So I'll focus on some applications related to fermentation, monitoring, diagnostics, food safety, and characterization of drugs. And the sensors that I'll be applying will be nanobead-based agglutination assays, nanograph substrates for plasmonics, and resonators just a little bit for degradation studies and for spectroscopy. And the research that I'll show you is performed in our group that we call the Nanoprobes team. It's based at DTU, Technical University of Denmark, just north of Copenhagen. And I'll acknowledge the people as we go along. So we've for many years now been working on sensors. It started with cantilever-based sensors, and then other things were added to that. We also work on centrifugal microfluidics. I'll come back to that. And then we work recently also on microfabricated structures for oral drug delivery. And that I will not cover today. But since we're working on oral drug delivery, we have a lot of new materials moving into our labs. New polymers, new drugs. And then on the other hand, we have for many years been working with nanosensors. So we now have a unique opportunity to kind of make a happy marriage between the two. And I'll show you some examples where we try to do that. So a lot of these new materials and drugs, they have needs for very specific characterization, where we can apply our nanosensors. And on the other hand, these nanosensors, some of them are developed without having one specific application in mind, so we can get a lot of help from the development of new drug delivery systems. And the happy marriage here in between is also facilitated by a grant from the European Research Council. The whole activity we call IDUN. It's named after one of our Nordic goddesses. And you can see here what we believe in. You can also follow us if you like. So we have a Facebook site and a LinkedIn site. And it's a collaboration between us, DTU Informatics, and then University of Copenhagen Pharmaceutical Sciences. So to the outline of this talk, I'll introduce you to discs if you're not familiar with them. I'll then move on to agglutination assays, nanoplasmonics using pillars, and in the end a little bit on resonators. So first, the discs and centrifugal microfluidics. For me, this started 9 years ago when I met Edwin here in the front. At that time, he was employed by Academia Sinica in Taiwan. And when I met him, he had just built a very cheap atomic force microscope using the pickup head from a DVD player to read out minute movements of an AFM cantilever. And at that time, we were working a lot with cantilever-based sensing, so I could see, wow, if he can read out a single cantilever, he can also read out 1,000, and then we can place them on a rotating disc, and we can have high throughput. So that's how it started for us. If you take now a Blu-ray player, if you still have one, you have one? Not many, you see. Outdated. But if you took it apart, it would have 3 lasers inside, 3 different wavelengths, all with a spot size of less than 1 micron, and with the capability of measuring deflections or height differences of less than 1 nanometer. It also comes with very fine mechanics. So you can rotate the disc very fast, several thousand RPMs. And also the pickup head is placed on a translational stage that you can use and repurpose as you like. And it all comes at a really low cost today. So, I moved on. So if you just imagine that you can spin a disc fast, what can happen? This is called centrifugal microfluidics. So if you make a DVD disc and you integrate fluidic channels on it, you can just use the forces that arise when you do the centrifugation to move your liquid around. You can do valving, you can do mixing, you can do filtering just by the simple liquid operations. And you don't need any external pumping. It all comes just from spinning. So in order to see what's happening if you spin a disc really fast, you need to build some sort of a test stand. So this is what we did. You can see a little motor here where you can place your disc on top, and then you have a camera on top, and then you start spinning, and you illuminate the disc with stroboscopic light tuned to the rotational frequency of your disc. So the videos I'm going to show you, it looks like the disc is standing still, but it's just because you're only seeing 1 frame per rotation. So if you just take one example, this one is a disc seen from the top with some channels inside, and we've just loaded 1 drop of blood in the center. Then we start to rotate, And what you can see is that the blood is moved outwards. And then you can see the plasma is starting to separate out. All the heavy components in the blood, they are sediment in the bottom of the chamber, and the plasma is on the top. And after 2.5, 3 minutes, you have a complete separation. Then you slow down the spin speed, and air that was compressed up here is now allowed to expand, and it pushes a little bit of your blood plasma around this corner up here and into this chamber where you have loaded your sensors. And this could be nanobeads for agglutination, for example. So the disc fabrication itself is easy. You can do it just by laser ablation or milling, or you can also, if you know the design you would like to use, you can make a master in nickel, and then you can do injection molding, quite similar to what they do in DVD manufacturing. And then you can get thousands of discs in no time. And then you integrate your little nanosensor, and you put a lid on top by bonding or gluing, for example. And that's the work of Marco here. So what type of sensors have we integrated so far? We have been integrating electrodes for doing electrochemistry on a spinning disk. We have been doing agglutination assays, resonator readout, cantilever-based sensing, and also integrating nanopillar substrates for plasmonic sensing. And today I'll show you examples of 3 of these integrations, the ones that I've highlighted here. So first, the bead applications, which is very much into diagnostics. So here we are utilizing small beads loaded on one of our disks, and then we're measuring how these beads are agglutinating. And then we want to measure the size of these clusters that are being formed. And the size is actually measured by just using the blue laser from a Blu-ray player and shining it through a small sample volume. And as we do that, the light will be modulated, and by this modulation of the light, we can then deduct the size of our clusters. If we move a little further and look at what's happening, we just have normally beads free in solution, and they can move around freely as they like. Then if we add a biomarker, they are going to bind, they're going to agglutinate. So now you can speed up this agglutination by applying a strong external magnetic field. And now they're bigger clusters. And if we now try to make them move by applying a very small external varying magnetic field, they're going to respond slower to that varying field because they're bigger. And it's this delay in response to the external varying magnetic field that we're using for our readout of this device. So we have tried with a lot of different biomarkers to detect. Either we buy commercially available beads for agglutination, or we functionalize our own beads and demonstrate it, for example, for this acute phase protein called CRP, or for other proteins available from our collaborators, and also biomarkers for dengue fever and Zika. And it works for all of them in the whole clinical relevant range, and it also works in full blood and from patients. So when we saw this, the previous pictures here of these young guys who are part of this work, they spun out a company, and it's called Blue Sense Diagnostics. I'll get back here to the next slide. So this is an early prototype from when this was still only in our lab. You can see every part is 3D printed, and you insert the disc on top, and there's a little bit of electronics to control our pickup head. And then you can see the whole flow of the measurements here. So you start by loading a small sample of blood on the disc, and then you insert the disc into the little reader unit, the motor, and then you start the centrifugation to separate the blood plasma, just like I showed you before. And then after 2.5, 3 minutes, you have a complete separation. You transfer a little bit of your blood plasma to the chamber with the magnetic beads up here, and then you incubate for a little while. You turn on the blue laser to do the measurements, and then you can get out a cluster size of your beads that have agglutinated, and then you have the response. So depending on the biomarkers you're measuring, it takes between 3 and 10 minutes to do the full analysis. So then you can imagine, instead of just looking for one biomarker in blood, maybe you would like to look for 2 or 10. And then you can imagine that you have different disks preloaded with different magnetic beads that will specifically detect whatever you're looking for. So the spin-out company Bluesense, they are now 20 people in Copenhagen and 10 in Taiwan. They have moved away from working on entire disks. They work more on pizza slices because this is a way to reduce price. And then each slice of a disk is then loaded with one specific type of nanobeads. And then you insert these pizza slices into a disk-formed holder and you can do your measurements. What they're targeting is mosquito-borne diseases like dengue and Zika, which is unfortunately spreading and becoming a wider and wider problem. And they just recently got a grant from USAID and now also just today a large grant from EU. So I really hope that they will be successful in bringing this device to the market. So when we had this transferring out of our lab, we still kept the technology in-house, and we're now using this for more fundamental studies. And this is one example. It's a collaboration with Graham Rina from University of Dundee, and then Xu Ling and Rokan from our lab. And here, the idea is that Graham has for many years been studying drugs that have been on the market for many, many years, like metformin. Metformin is a type 2 diabetes drug, and still is not fully understood how it's actually working. One of the hypotheses is that metformin will go into our cells, into the mitochondria, bind copper, and transport copper out of the cells. And in this way, it's regulating the energy production in the cells, specifically in the liver. So Graham would like to test this hypothesis. So we did something rather simple. We just took some beads. We started with some micron-sized beads, so we can actually just see them in a microscope. We functionalized them with cysteine, which is known to bind copper in the mitochondria already. And then we could look just in a microscope. Yes, the beads are nicely suspended. Then we added copper. And we expected to see strong agglutination because cysteine likes to bind the copper. And this is certainly also what we saw. The beads are clustered. And then when we add metformin, we can break these bonds again and go back to suspended beads in solution. This is a very simple way of analyzing copper binding. So then you can continue and you can just play around with the dose of metformin that you're adding. So you can actually see how different doses of metformin brings out more and more copper. And you can also get other drug analogs from Graham and test those in the same way. So why do we do this on the disk? Because we do all these experiments on a rotating disk. It's simply because all the fluidic handling is much easier. And secondly, we both have mounted the optomagnetic readout, so the blue ray blaze sensing. And we also have mounted like a 3D imaging system. It's like a confocal microscope that can count individual particles or cells down to a size of 1 micron. So we have both options for sensing. And it also means now that Rokan is working on combining the optomagnetic readout for detecting small biomarkers using the nanobeads, and at the same time counting and monitoring the amount of white blood cells in a blood sample. So you have combining two sensors and you get additional information out from your blood sample. So that's work in progress. So much about nanobeads. I'll now move on to nanograss, as we call it. It's a substrate for doing surface-enhanced Raman scattering. So in Raman scattering, are any of you working with Raman? Okay. So in Raman scattering, you're relying on a very weak effect. In normal Raman scattering, you would shine a laser onto a substrate, and once in like a billionth times, the photon will be inelastically scattered in your material, and the wavelength that comes back from your surface is slightly shifted. And this shift that we are looking at, but it's a really weak effect. So since the 70s, people have discovered that if you place, I'm doing it again, if you place these molecules on a roughened metallic surface, for example, silver or gold, you can increase the Raman scattering. And this is called surface-enhanced Raman scattering. So our first task was try to make these rough metal surfaces. And our approach, first started by Michael and later explored also by Caillou and Thomas, is to etch by a maskless technology called reactive ion etching into silicon. And then you can form nanopillars or nanograss. And then afterwards you coat it with silver or gold, and then you're ready to measure. And the surface, it looks like this. And you can do it on 4-inch or 6-inch wafers. And it's a very simple etching process. And you can tune the height of the pillars by the etching time and the aspect ratio by changing the gases in the etch chamber. Then when you have finished the etching, you have a wafer. When it comes out of the clean room, it's more or less black. And then you can coat it. In this case, it's coated with silver. Then you cut the wafer into small pieces, a few millimeters by a few millimeters. And then you're ready to do your measurements. You just put the analyte on top. And you can ship it around to collaborators if you like. So when we started these substrates, we discovered that they're enhancing a lot, much more than we had anticipated. And when we looked into it, we learned something rather interesting. These pillars, they're flexible. So if you add a liquid on top and you let the liquid evaporate, you will actually form small clusters on the surface because the forces generated when the liquid evaporates will pull these pillars together. And this is now an ideal configuration for surface-enhanced Raman scattering. If you just zoom in on two of these pillars, first you bring in your analyte, just from air or from solution, and then you bring the heads together. These heads are metallic heads, and they are called electromagnetic hotspots. So this is the ideal configuration for surface-enhanced Raman scattering. And this is just the SEM image when they have been leaned, and you can see how they are leaning together with the metallized heads. So we wanted to check some years back, is this really working? And we just worked first with a molecule called thiophenol, which is known to have a very strong Raman signal. And we could see if we just bought some commercial substrates, so that's the red curve here. This is the signal we get. Here is our shift in Raman signal, and here we have the intensity of the signal. And then the same goes for our non-lean pillars, that's the green curve. Then if we lean the pillars together and do exactly the same measurement, our enhancement is now 700 times better than from the commercial substrate. And if we pre-lean and then add our molecules, we are still like 100 times better. So it really works with this leaning. So this is also why a few years ago a small company was spun out where researchers can just buy and use the substrates for whatever purpose they have. We've continued since then we started with silicon structures, then moved on to doing similar structures in glass and in polymers, and also recently making nanohudos on top of which these metal heads can actually move around and make local hotspots. But since we now have this centrifugal platform, why not integrate these substrates onto the platform? And there are many good reasons for doing that, and I'll share with you today two of them. So you can first of all integrate a lot of fluidics together with your substrates. And secondly, you can use the substrate itself as a way of filtering your sample. So first, the integration of microfluidics, and here I'll take an example from the development or production of drug compounds. So to do new drug compounds, you can do oil-based chemistry, you can work directly with elements from nature, or you can do biofermentation. And biofermentation is really popular in Denmark. We have a pretty large pharmaceutical industry. So if you try to convince microorganisms to produce chemicals for you, it's really a challenge to figure out which ones to pick, which ones are the winners, which ones are producing the most, and will they continuously produce over time. So there's a need for sensors that can actually monitor this. So in Denmark, we have a lot of biofermentation for the production of insulin and growth hormones, but also in general just for a lot of chemicals going into production of tablets. So if you try to visit some of these labs where they do biofermentation, or if you go to the big scale industry, I think you'll be shocked when you look at these fermentation tanks because it's really dirty. This is what it looks like. Inside you have your producing organisms, you have all the nutritions, they even add antibiotics to this. So you want to sense a very small molecule in this very complex matrix. And you want to see, do I have a low production or a high production, and how is this going over time? So we got a producing organism from our collaborators from DTU Biosustain. This is a gene-modified E. coli that will actually transfer tyrosine, which is an amino acid readily available, into a high-end chemical called picoumeric acid. So we got that one from them, and we got the cell supernatant from our collaborators. And you can see here, we in the beginning just tried to centrifuge our samples, sediment out all the cellular components, and then dilute the samples a little bit, put them on a SAS substrates, and tried to measure. And what did we get? We didn't get a thing, because everything is, hidden in whatever background there is in the sample, and on top of that, the signals from tyrosine and from picumeric acid. Tyrosine, that's what we feed the organisms with, and picumeric acid is the producing thing. They overlap. All the peaks are kind of overlapping, so we can't see anything. So we needed to rethink. So we introduced a simple sample pretreatment step. It's called liquid-liquid extraction. So you put your cell supernatant in an acidic environment such that the picumeric acid will be neutrally charged. Below you have an organic solvent. And now the neutrally charged molecules can actually go into the organic solvent and stay there. And now we just take out a little sample from the organic solvent and put that on our SAS substrate. And now, suddenly, we can actually see a clear peak from the picumeric acid and nothing from the tyrosine. And this peak we're then using to make calibration curves so that we can actually start doing quantitative measurements in cell supernatants. And now we were then ready to actually get real producing strains and real cell supernatants from different strains. And we were able actually to measure directly on the SAS substrate after the liquid-liquid extraction the amount of picumeric acid being produced. And it compared very well with HPLC data that our collaborators were doing on exactly the same cell supernatants. So our measurements are fast. They take around half an hour compared to HPLC, which takes long time to collect the samples. But it's still rather manual because you need to do this liquid-liquid extraction. But of course, with all our disks in-house, we integrated all the steps, the filtration, metering, the liquid-liquid extraction itself, and the SAS measurements onto a disk platform. So that's the work of Laura and Lydia from the previous slide. So everything can actually be integrated in a centrifugal platform for this type of measurements. So finally, Onur, he didn't do lab on disk. He moved on and realized that our SAS substrates themselves are interesting filters. He's working, for example, in milk, another complex sample, and then we spike it with food adulterants like melamine. And you can see milk itself is very complex, so it's hard to measure directly in milk. But you can actually use our SAS chips for the filtering step as well. And you can see here we have integrated SAS chips directly on our centrifugal platform. What's happening is that when we introduce the milk sample that has been diluted to the SAS chip, it's placed here. You're looking now from above on the disk that's rotating. You can see that we are only covering part of the SAS chip. This part up here is kind of dry. But what we realized is that liquid is moving upwards on the SAS chip, much like in a filtered paper. And if you now zoom in, you'll see that this region here is very dirty, even after we have dried it. So you have a lot of leftovers of proteins here, whereas this region up here is super clean. If we then look at the SAS signal, you'll see that you have a very clean signal up here, where you have completely purified your sample. Only the melamine peak is still left. Down here you have a very contaminated region. So the SAS chip itself, with all its nano pillars, works as a nice filtering paper. And here you can see it a little further, how it actually travels upwards. Good. And then by using this clean region above, we can actually do again a calibration curve and detect melamine in milk directly in the milk down to a few ppm. So you can ask, why do you use the centrifugation and the disk? Couldn't you just dip these small SAS chips directly into the milk? Yes, you can. We've also tried. But in that case, you're actually 70 times better if you do it on the disk. And this has to do with the way that we're actually controlling the liquid and only producing a very thin sheet of liquid moving on top of your SAS substrates. So I have a few seconds left. I just mentioned very briefly our resonator work. It has already been introduced by Michael, how we can use small nano and micromechanical resonators for a lot of different applications. The thing is that these are very sensitive to mass changes and to temperature changes. So if you take a cantilever that's resonating and you coat it with a polymer, the resonance frequency is going to detune, it's going to be lower because you have added mass. And then if you try to degrade this polymer that's coated on top, you can actually follow how the resonance frequency is going back to its initial resonance frequency. And this way you can get a very precise measurement of minute changes in mass changes as you're degrading a polymer, for example. But it's still just a one cantilever, one resonator measurement. So what we could do, inspired by our Blu-ray work and Taiwan again, was simply just to build a system where we're using the translational stage from a DVD player, and you have a Blu-ray pickup head below. And inside you have a liquid chamber, and you have 20 resonators sitting there in parallel. And then you're simply running different degradation media through. All the resonators are coated with a polymer that we use for drug delivery. And then we can simply study the degradation over time. And this is an example where we can then study degradation of a polymer called PDLDA for different enzyme concentrations. And compared to what you'll see in literature when people do degradation studies, we can do this within hours instead of 6 weeks that's reported. And that's simply because we just have a much more sensitive method, and we also have 20 resonators that we can compare to each other. So much more reliable data can come out of this. I think I'll skip my last example related to spectroscopy due to time, and I'll just move on to saying that I hope that you can see that you can actually repurpose technologies that are already available out there. They're really super sensitive, and there are kind of a lot of new things you can do if you open them and play with them. I've shown you examples within fermentation, diagnostics, food control, and characterization of drugs. These are the sensors that I've shown you, and thanks to our different sponsors. And also I just say that we're running 2 PhD schools each summer in August, and you're very welcome to come yourself or send your PhD students. They will be working hands-on with some of the technologies I've shown you. There's both one in sensors and one in drug delivery. So thanks for your attention.